Hi everyone, welcome to this M2D2 talk. Our speaker today is Jan Smith. He's a PhD student at the University of Toronto in the lab of Benjamin Hebskens. The lab studies pharmacogenomics and radiomics for cancer research. And before joining, the University of Toronto, Jan worked as a computational biologist at the cancer program at the Broad Institute. Thank you so much, Ian, for accepting to present your work to this crowd today, and looking forward to it. The first yours. And before, before we start, everyone, if you have any question, please raise your hand or just, and whenever the speaker feel like it, he will just let you ask your question or ask your question in the chat, and I'll be more than happy to repeat that for the speaker and get you answered. Thanks, everyone. And Jan, the floor is yours.
Thank you very much, Prudencio, and thank you to you and the M2D2 talk for this opportunity to talk about my research. Certainly, it's always exciting when people take an interest in the problems you're trying to solve. I'm dealing with a little bit of a cough, so I'll do my best to not cough into the mic and keep restrained. So the title of my talk is Learning Representations on Biological Data with Weakly Supervised Learning. And so a little bit of context, the broad direction of my research and a lot of these problems is precision medicine. That is, the goal is to, especially for something like cancer, optimize the treatment for a patient based on the specific molecular features of a patient's disease. A lot of studies have shown, clinical trials have shown that when you can match a therapeutic agent to a patient based on some molecular indication or some indication of efficacy, the therapeutic treatment is more likely to be effective at treating the patient's disease. So this is a nice goal rather than a one-size-fits-all approach where you treat everyone with the same standard of care. There are two major challenges to precision medicine broadly. First, you have to be able to identify biomarkers or molecular indications of drug efficacy, and you need drugs. And the reality is that there are many more potential cancer vulnerabilities like oncogenes that we would like to be able to target with therapeutic agents that we simply can't because we don't have the candidate drugs. I would like to highlight recently KRAS, which was thought to be undruggable. Two KRAS inhibitors, sotorasib and adagrasib, have been developed. I don't actually know how long it took to do that, but it's a very exciting development in the field of cancer therapeutics. So I'm mostly going to talk about the drug development problem, which I actually think there's a talk later today about drug development. So developing new therapeutics is a really difficult process, typically requiring 15 or more years from going from target identification and preclinical study to FDA approval. And development of a new therapeutic is estimated to cost over $2 billion, which in large part due to the failure of candidates in the drug pipeline. And the principal reasons for these candidates failing are lack of efficacy or safety considerations like unintended side effects. There are a lot of reasons for this bottleneck, but the crux of the matter is we're just not very good at going from candidates and identifying drugs that can be, or candidate drugs that can be developed into therapeutic agents. And while we're trying to do this, there are a lot of patients that simply don't have therapeutics that can be used to treat their disease. So we have both a medical and an economic imperative to improve the efficiency of drug development. So there are two kind of typical approaches you might use to identify therapeutics. First, you can start from I don't know if you guys can see my mouse. You can start from a disease-relevant target of interest. So say you have a protein that you know is relevant to a particular disease, and you want to find a new therapeutic agent that will inhibit it or bind to it. So you can then screen with, say, a binding assay against some library of small molecules, of which many exist with 10 to the 5 different candidate therapeutics. This is difficult because it doesn't scale all that well. Protein targets are frequently mutated, and so you might have to do this sort of procedure for each mutant protein of interest. And even still, there's no guarantee that something that works in a model system or a binding assay is going to be relevant in a patient. So one alternative is to do sort of a data-driven approach where you characterize the function of each molecule in your library and interrogate its function using phenotypic screening. So phenotypic screening is designed to be a rapid, high-throughput, inexpensive method for characterizing function. So the basic idea is you take some model system like a cancer cell line, you treat it with your agent of interest, which could be a drug, could be some sort of a genetic inhibitor like a CRISPR silencing of a gene, or even a disease, and you measure some set of features in whatever data modality you happen to be interested in. You then compare this to a controlled experiment where you absent the treatment, and take the difference between these two measurements to arrive at a differential phenotypic signature. And the feature spaces that have been most commonly used for this kind of analysis are gene expression, cellular morphology, so the shape of cells, viability, where each feature might be whether or not the drug kills a particular cell line, and you measure in many different cell lines, or proteomics. So there are a lot of different technologies for doing this, and really what enables it is being able to do it fast and at scale. So for a lot of these experiments, a phenotypic screen of a compound that you don't know the function of might cost $5 to $10. And so you can do that at massive scale, generate candidate hypotheses, and then try to, given that insight, do more intensive validation of the candidate. Sorry, it's not, okay. So these, perturbational data sets measure these changes in features. And recently in the past, 5 to 10 years, a number of public data sets have been generated. Two that I really want to highlight. First, the L1000 dataset, which measures changes in gene expression. It was originally published in 2017 from the Broad. And yeah, it uses a reduced representation of the transcriptome, measuring only 1,000 genes, which is cheaper but more scalable, to sort of characterize the function of these perturbations. Similarly, the cell painting morphological assay, also from the Broad Institute, from Ann Carpenter's lab, uses cancer cell lines as a model system, treats them with perturbations, and measures changes in cellular morphology to characterize function. For concreteness, this slide shows a typical workflow of a phenotypic screen. You begin with some model system like a cancer cell line, treat them with the perturbations that you're interested in, ideally a large number of them. And then you measure the set of features. So for example, in cell painting, you treat them with fluorescent stains. The stains bind to different organelles in the cell, and you can image the cells with microscopy. Just take a lot of pictures of a lot of these cells that have been treated, calculate the features, get a differential feature profile or a signature of each perturbation, and then do some sort of downstream analysis, like try to identify perturbations that are related to each other. The general, there are a lot of things you can do once you've generated one of these big data sets, but the sort of basic analysis requires computing similarities. So you can imagine you have some perturbational data set where you have differential features, and each one of these columns is a different signature or experiment. You then take a similarity function and compute the pairwise similarity between all pairs of signatures in this data set and get this big similarity matrix. And the density of the similarity matrix might look something like this. The underlying assumption, assuming you've chosen a relevant feature space, is that if you have an unknown perturbation that is similar to a known drug or similar to something whose function you can make a hypothesis about the mechanism of action of the unknown perturbation and do it much more rapidly and cheaply than some more in-depth experiments. But this raises an interesting question. What is a good similarity function? What does it mean for something to be similar in one of these biological spaces? So the tool for perturbational analysis is a similarity function. And some common examples that have been used in the literature over the years are correlation measures like Pearson and Spearman, where you just take two vectors and ask if they're correlated. For gene expression, there are a number of gene set methods like GSEA and GSVA. And my personal favorite is cosine similarity, which is very closely related to Pearson correlation. Now, these similarity functions are usually scale invariant, which means if you multiply a signature by a positive constant, it doesn't change the similarity. And this is important because different perturbations that do the same thing might have different potencies. And so if you have, especially if you're measuring them at a range of doses, time points, and so on. And so if you have two perturbations that are doing the same thing phenotypically, but to different degrees, you're actually more interested in the direction of the changes than the absolute magnitude. So all of these examples have this nice scale invariant property where we're interested in the direction of the phenotypic changes rather than the magnitudes. So a good similarity function then is one that can correctly identify biological associations from related pairs of signatures. This is challenging because we don't always know what the relationships are. If we knew what pairs were biologically related, we wouldn't have to do the experiments. And the big challenge in all of these data modalities is that high-dimensional biological data are quite noisy. The signal-to-noise ratio is poor. So this is an example from L1000. We're looking at pairwise similarities between signatures in A375, which is a melanoma cell line. So the distribution of cosine similarity between everything in the database is in green, and the similarities between replicate experiments, which should be producing the same answer, is in gold. And as you can see, this ground truth of replicate similarities is not dramatically different from the background. It's really hard to discriminate because of all the noise and the relatively weak signal. This is another example. We're looking at a UMAP visualization of viability measured with the PRISM assay from Corsello et al. And so each one of these points represents a different drug, and the space of features is whether or not the drug killed a particular cell or cell line. And you can see that some compound classes discriminate themselves quite well, like bromodomain inhibitors and EGFR inhibitors, say, are well discriminated in this experiment. But there's this giant blob of compounds that, with a lot of annotated classes from prior knowledge that are not good at discriminating themselves in the background because of this noise problem. So then this motivates the hypothesis that we want to learn an appropriate representation on these data, and that doing so will help us detect biological associations. To explore this, I'm presenting two works. First, sort of a more theoretically motivated work characterizing the distribution of cosine similarity for covariate data. And second, using weakly supervised learning to learn a similarity function from the data directly. So the first work is titled On the Distribution of Cosine Similarity with Application of Biology. It's available on arXiv, and I collaborated with a number of people from my lab. The sort of underlying motivation behind this is that biological data has complex covariate structure. If you have an example perturbational data set, or even like an scRNA data set, there's going to be a lot of covariance. And this manifests in both covariance among the features and covariance among the samples. So feature covariance you get because when you're measuring two different features, they're not actually independent. So for example, two genes in the same pathway are likely to have some correlation if they're being modulated by a common transcription factor. Sample covariance is a little bit more, well, we understand it because these samples aren't drawn IID from some distribution, but they're compound classes or genetic inhibitors, and so a lot of them happen to be related to each other. But the feature covariance is what's especially interesting. So to state the problem that I'm trying to solve, the goal is to estimate the probability distribution of cosine similarity given a data distribution. So imagine you have data living in an n-dimensional space. This is a little cartoon kind of showing some density plot, and you can characterize this data distribution by the mean and covariance of your data. If you then take samples from this data set, IID, and compute cosine similarity on those samples, you're going to get some distribution. And you can characterize this distribution with the mean cosine similarity and the variance or standard deviation of the cosine similarity. And the problem, or what we want to do, is come up with some sort of functional form, like write down what the mean and variance of cosine similarity are as a function of the mean and covariance of the data distribution. So we'd like to be able to say, okay, what is the mean of this cosine similarity and what is the variance of that? And this covariance structure is really important because of the manifold hypothesis, which is prevalent in high dimensional data and especially biological data, where, say we're measuring 1,000 features, like 1,000 genes and their expression. The data doesn't actually live in a 1,000-dimensional space, but in fact lives on a manifold that is typically a lower dimension than the feature space that we're measuring. And so because it lives on that manifold, there's this complex covariance structure where many of the axes that you're measuring are not actually varying independently of each other. I'll get into a little bit more about why we're motivated to do this, but it kind of started out as a sort of interesting math question. So cosine similarity, I imagine most people are familiar with, but just in case you're not, cosine similarity is a normalized inner product. So if you have two vectors A and B, it's the inner product of A and B, so A transpose times B, divided by the norm of A and norm of B. And it has a lot of nice mathematical properties. Its values are between -1 and 1, where 1 corresponds to completely vectors that are pointing in the same direction, and -1 corresponds to vectors that are pointing in different directions. And this is an example distribution of cosine similarities on perturbational data. So to provide a little bit of motivation for why we care about this, I want to highlight how the covariance structure of data can affect discrimination. So consider a simulated data set. This was just an in silico experiment where you have a 100-dimensional space and you have two classes. So class 1 has some mean and covariance structure, and there's some background distribution, which is mean 0 and the same covariance structure. And this cartoon depicts what this might look like, where you have this one class in blue and this background distribution in red. So with perturbational profiling or phenotypic screening, you want to be able to identify pairs of points in this class 1 based on their similarity profile. So you could imagine you compute the cosine similarity between all your data points, and this red distribution corresponds to data points from class 1, and this blue distribution corresponds to the similarities of everything. You want to be able to discriminate this red distribution from this blue distribution. To illustrate why this can depend so much on the covariance structure, I introduced two linear transformations on the space that modify the underlying covariance. And you get distributions that look like this. I did this intelligently. I had some goals in mind, but this is just a linear transformation on the simulated data. And what you can see is that this transformation 1, it's much easier to separate the red from the blue distribution, whereas in transformation 2, they're essentially overlapping. And you can quantify the statistical power at a particular alpha if you rank red relative to blue. And as you can see, there's a massive improvement in power if you choose an intelligent transformation on your space. And this is just a linear transformation where we're modifying the underlying covariance structure of the data. So this sort of cute little example suggests that if we can intelligently choose representations, we'll be better powered to identify associations. Okay, so cosine similarity. One of the first things you can observe is that the variance of cosine varies with the dimension of the data space. To illustrate this, suppose you have some data that's just a bunch of standard normals. So these are all independent IID, mean 0, variance 1, normally distributed values. You take pairs of vectors from this space and compute the cosine similarity between them. You can then ask, okay, well, what's the variance of this distribution as the number of features, the dimension of the space changes? And this is a simulation where we look at 3 different dimensions. For a 10-dimensional space, you get this big fat red distribution, 100-dimensional spaces in blue, and 1,000-dimensional spaces in green. And you can see that as you increase the dimensionality of the space, the variance of cosine shrinks. This is a phenomenon closely related to the cursive dimensionality, or something like VC dimension, that as you add more features, everything starts to look you kind of shatter your space. It's more and more dissimilar or less related to each other. The probability of seeing two vectors pointing in the same direction in really high dimensional spaces falls off quickly. So I then want to just get into some results from the work that we did. The simplest example of data is if you have n-dimensional data with mean 0 and identity covariance. So this is analogous to this example where you have IID components with normal 0, 1. And this cartoon kind of shows what it looks like. You have a nice, spherically symmetric Gaussian, and this is equivalent to a uniform distribution on the unit sphere. So for this data, you can ask, what is the variance of cosine similarity if you sample points of the distribution, and you get this result. So the dimension here is on the x-axis. We're looking at a log plot. The standard deviation of cosine similarity is on the y-axis. Red is the theoretical value that we calculate, and blue is empirical. And it's relatively easy to show that the variance of cosine similarity goes as 1 over n, and there's essentially perfect agreement between experiment and theory. So this is, quite maybe intuitive given that this example. I don't think this particular result is all that surprising. You see it with Pearson correlation also. So, okay, things get a little bit more interesting with a more complicated data case. So imagine you have a data distribution with some covariance matrix with, now the eigenvalues aren't all equal. So here's a little cartoon where you see that the axis that's longer has a larger eigenvalue than the smaller one. But we're still mean 0. So this is a distribution centered on the origin. It's not necessarily normally distributed. I think this particular graphic was, but so we have non-zero covariance. Again, you can ask, what is the mean invariance of cosine similarity for a distribution like this? And you get this elegant looking result where the mean of the cosine similarity for data drawn from this distribution is 0. And the variance is this, concise function of the eigenvalues. So you can see very clearly that the variance of cosine similarity intimately depends on the eigenvalues of your covariance matrix. And this is a simulation where we sampled data from a variety of eigenvalue distributions. The theoretical variance, according to this formula, is here on the x-axis, and the empirical variance, where you just sample points from this distribution, calculate cosine similarity, and calculate the variance, is on the y-axis. And as you can see, there's very good agreement. This is an asymptotic approximation, so it does break down somewhat as the dimension gets small, but it still does a pretty good job capturing the variance of cosine similarity. And finally, the most general case, you have data with some covariance matrix with non-zero eigenvalues and not necessarily equal eigenvalues, and a non-zero mean. So it might look something like this. Again, not necessarily Gaussian distributed, just a general data distribution with some properties, some assumptions. You can then ask, what is the variance of cosine similarity? And you get this, slightly more complicated, but still elegant result, where we can write down the mean of cosine as a function of the data and the variance of cosine as a function of the data. Again, we simulate it. This gets a little bit trickier because the asymptotic approximation breaks down more as the mean tends toward 1, as the cosine similarity distribution tends towards 1. But we still see generally good agreement for an empirical validation. And honestly, this is, given that I work in biology, this is one of the more aesthetically pleasing results. I mean, you could print this on a t-shirt, and I'm not sure anyone would really know what you were talking about, but it's a very elegant relationship between similarity. Yes, a question. So does the distribution of cosine similarities depend only on the mean and variance for non-Gaussian distributions? No, but the moments do. So the mean and variance of cosine depends only on the mean and variance of the data distribution. With some assumptions, you need a finite fourth moment of your data. But if your data is compact, it satisfies all the assumptions. If you're dealing with some sort of diabolical distribution, then that might break down. Okay, so a couple Pudestia?
Yes, following up on Jason's point, what happened when you have multi-mode distribution? You have different modes in the distribution.
That's a good question. I think that again, the mean and variance of cosine should still be given by these expressions. Like, I don't think it assumes unimodality, but obviously the shape of cosine similarity could be multimodal in turn.
Okay, thank you.
Okay, so this was an interesting math problem, but why should we sort of care about this? One result that's kind of interesting, if you look at this, the case when you have centered data with some covariance, we have this nice expression for the variance of this distribution. It turns out the variance of cosine is minimized when all the eigenvalues are equal for centered data. So this result where the variance 1 is 1 over n actually turns out to be a lower bound on the variance of cosine similarity. Basically, any other representation of the data is going to be fatter. The distribution of cosine similarity is going to be fatter. I describe this result as sort of an isotropic principle that we can minimize the variance of cosine similarity when all the eigenvalues are equal. With non-zero mean, it turns out to be slightly more complicated, but still nearly isotropic. The eigenvalues differ by no more than a factor of 2 when you're minimizing the variance. Now, the reason this is interesting, again, we're trying to discriminate these two distributions. We have something that we think is biologically related or similar, like this gold distribution, and we have this background in blue. Now, low signal to noise means it's going to be really hard for us to move this gold distribution very far to the right because the noise is just so strong. But if instead we can learn a representation where we minimize the variance of the background, it becomes easier to tell things apart. And maybe to add a little bit of intuition, this is kind of like when the variance of cosine similarity is really large, say in a 1,000-dimensional space, that's because the signal is being dominated by a small number of eigenvalues, a small number of signals. And so if you can bring all of the signals more into equilibrium, which is this isotropic concept, then all of them are contributing kind of equally. Now, okay, so there's some interesting applications of this. I mean, it's kind of a beautiful result. And to my knowledge, I don't think anyone's done this before, although it's possible that there's some 1960s statistic paper where somebody did this. So a couple of things you can do with this. First, you can estimate an analytical null of cosine similarity given the properties of data. Typically, people now compute a background distribution with cosine similarity and use that as their null, when in fact, it could be biased, for example, because of sample covariance. But if we know the mean and covariance structure of the underlying features, we can arrive at an analytical null. Secondly, as sort of motivated by how transformations can improve statistical power, we can intelligently choose embeddings or transformations to identify similar elements, which is what we want to do with perturbational data. And this provides kind of a theoretical motivation for exploring the space of embeddings that, a simple linear transformation on your data can change the covariance structure in an interesting way. Sorry, was there a question? Okay, just background. I'm sorry, I can't hear you very clearly. Maybe you could type your question in chat.
Thank you to the great community for.
Okay, could just be background.
Yeah, absolutely.
So to sort of look at this, the second point, intelligently choosing embeddings, we can use this result to kind of optimize statistical metrics over some space of transformations. So suppose, simplest example, you have data with two classes, illustrated by this sort of graphic. If you compute cosine similarity on your data, you get this result. And we can write down what the statistical power is at some alpha or whatever metric you happen to like a signal to noise ratio, as a function of the properties of the data, and then choose embeddings to maximize the power as a function of these input parameters. Now, this turns out to be a little difficult to do in general. I've only really explored the two-class problem. But in principle, you can sort of analytically do this. The challenge is you have to know the mean and covariance of your data classes. And in perturbational experiments, you are kind of assumed not to. Like, you don't necessarily know what where the data happens to lie for these different classes. I also just want to mention that there have been a number of papers, usually motivated by a confounder, that have employed transformations like this one, or like the ones we explore, to improve signal and remove confounders. And so, these were always motivated by some anomaly that people observed, but we can sort of explore the space of transformations more comprehensively than you can if you're just trying to remove one confounder. Okay, so that provided a little bit of theoretical motivation for doing these embeddings, but in practice, there's not really a method there for using it from the cosine distribution work. So the second work is a much more practical approach, and it's perturbational metric learning, or PeML. So the onset, excuse me, the underlying assumption behind doing metric learning is that the features that you measure are not necessarily the optimal basis for computing similarity of perturbations. So for example, suppose you're looking at transcriptomics. Gene space, mRNA, is relatively easy to measure. We've gotten very good at doing RNA sequencing. It's conceptually very, very tractable, very approachable. You're measuring the abundance of molecules in the cell. But the problem is that it weighs biological processes disproportionately. So for example, suppose you think about pathways as these biological processes. Cell cycle has a much larger impact on gene expression if you're just measuring all the genes compared to something like notch signaling, or some kinase pathway. That all of these signals are on different scales. And what we saw from the theory is that when they're all on different scales, some signals tend to dominate, and it's a lot harder to identify maybe the signals that are most important for discriminating perturbations. To illustrate this empirically, what I'm plotting here is the cumulative percent variance explained by the different principal components for perturbational data sets. So this green line is cell painting, the morphological data, and these other lines are different cell lines from the L1000 data set. And these kinds of plots are pretty common in papers. You can see that the different principal components, the different eigenvalues, have quite a dramatic range, up to several orders of magnitude, where the leading principal components just explain a colossal amount of the variance, and then everything else is really subtle and hard to discriminate. So the goal then is to learn an embedding to maximize our power to identify relationships. So most people here are probably familiar with self-supervised learning. If you're not, I'm just going to do a quick 30-second recap. Self-supervised learning is a really exciting technique there's been quite a revolution, especially in computer vision and NLP. The basic idea is to learn a representation of the input data directly from the data, but without labels. And so this is an example. This is SimCLR from Chen et al. From Jeff Hinton's group. The idea is you take an example data point, so say this picture of a dog. You apply two transformations that don't change the identity of the data point, and generate the synthetic augmentations of xi twiddle and xj twiddle. And then you learn a representation on the data that keeps these pairs of augmentations similar to each other. And this is really exciting because you don't actually need to know that this is a dog to know that these augmentations are also of dogs, because you have a set of transformations that do not change the identity of your data point. This is really powerful. The problem is you need identity preserving transformations. And in biology, we don't necessarily know what transformations on a point from gene expression won't change the identity of the compound that was used to treat it. So because we can't do self-supervised learning, this motivates weakly supervised learning, which is like the poor relation. So instead of using data augmentations that don't, these are still both pictures of a dog, we use replicates. The assumption is that if you repeat an experiment in a perturbational assay, you should get the same answer. Now, there are a lot of reasons why that might not be true. There could be technical artifacts like batch effects. The experiment could have been improperly done or failed for some QC reason. There are other reasons why this benchmark isn't perfect. Like a compound might not actually do anything in the feature space or the cell line or the dose that you're interested in which case you're just comparing noise. But we assume that that replicate pairs are similar, and this provides our notion of ground truth that we can use to train an embedding. So this illustrates this a little more clearly. We want to learn a similarity function to discriminate known related signatures from unrelated signatures or non-replicates. So for the similar case, we have two experiments that we repeated. So same cell context, same perturbation, and we compute the similarity between them using our similarity function and get some distribution that looks like this. And then we have, for the negative case, just two signatures whose we don't know a relationship to. They're not of the same perturbation, and this gives us a background distribution. We can then learn an embedding to make the similar replicates as similar as possible while keeping the background distribution dissimilar. And so the learn metric, like we take cosine, it looks a lot like cosine similarity because it's a parameterized version of it. Instead of just taking the inner product between A and B, we take the inner product between the learned representation of A and the learned representation of B. And the loss function that I use for PeML, although you could use a lot of different loss functions, but the loss is a T-loss where you look at the mean of the known similar pairs minus the mean of the dissimilar pairs divided by the standard deviation of the dissimilar pairs. So this borrows a little bit from the cosine theory. Everything I'm presenting in PeML is a completely general framework. You can take whatever architecture you want to use to learn this representation, feed-forward neural network or VAE or whatever you happen to like. The results that I'm going to show today are all linear transformations, and that's a little sacrilegious in 2023 when everything's deep learning. I'm certainly looking at deep learning architectures, but I think there's some interesting arguments for using a linear architecture. So just briefly, the first is simplicity. If we can find a simple representation like a linear transformation, if it gives improvement in our ability to reconstruct biological signal, then it may be useful, even if it's not complex. Secondly, in the literature, for example, in these papers, linear transformations were used to correct for artifacts that then resulted in improved downstream analysis. Another major concern is that, especially for transcriptomics, we might want to use our embedding and then take gene sets from other experiments and other papers, and so compare our embedding to out of distribution data. And it's not clear, but I have some concerns that something like a VAE, this out of distribution data might not be well represented in the representation. And finally, these aren't pixels in an image. The quantities that we're measuring should have some biological significance, like gene abundance or protein abundance or whatever you happen to be interested in. So staying close to the original representation has some advantages. Okay, so yeah, they're dual interpretations. I'm going to skip ahead a little bit in the interest of time. So the workflow for PeML, you take a data set, you take pairs of replicates, which you assume to be similar, and treat this as your similar group. You have pairs of non-replicates, which you assume to be unrelated, even though some of them might actually be related. And then you learn an embedding to bring replicate pairs close together and keep non-replicates far apart. And this is our similarity function. I represent it with this little blue arrow in a lot of the subsequent slides. This is implemented in Torch, and so it'd be relatively easy to substitute in alternate architectures, and we use mini batches where each batch corresponds to a compound. Okay, so we benchmark this method with two benchmarks, which should be our ground truth. Again, it's imperfect. First, we have replicates, which we use different replicates for training and evaluation. And second, we use pairs of compound signatures belonging to compounds with the same mechanism of action. Prudencio.
Yeah, I was just going to ask if by using the replicate, like the same biological replicate, or technical one, the technical replicate, is this method allow you to eliminate the batch effect if the technical replicate are from different batches? Or have you kind of used technical replicate from different batches?
Yeah, it's probably preferable to use replicates from different batches. I believe most of the replicate pairs are in fact from different batches. There's a paper that I'm going to reference at the very end that actually does try to do batch effect correction in the process of learning weakly supervised features. So I'll circle back to that. So in this work, I apply PeML to two different data sets from two modalities. First is L1000, has order 10 to the 5 signatures, order 10 to the 4 compounds in a variety of different cell lines in gene expression space. So you're measuring 1,000 differentially expressed genes. And the second is the cell painting morphological assay in U2OS cells with, again, order 10 to the 5 signatures, 10 to the 4, I believe it's 25,000 compounds, and a little more than 1,000 morphological features. So both of these are relatively large perturbational data sets. We can then use our benchmarks to assess performance. I compare with Cosine because it's kind of an untrained version of PeML. You start with for a benchmark, you have a set of similar signatures. You then compute the similarity of pairs of similar signatures using PeML and Cosine. This is just a plot for one of the metrics. You get a distribution of known similar relationships and a distribution of the background or all pairs. And then you can summarize this result with a couple different metrics like the AUROC, your statistical power at a particular FDR. Both of these are non-parametric evaluations of whether or not you can discriminate this gold distribution from this blue distribution. And you can also do a parametric summary statistic like the signal to noise ratio, where you're doing a better job if you can separate those distributions as much as possible. And the two benchmarks are, the first ground truth is replicate signatures, and the second ground truth is compounds with the same mechanism of action. And this ground truth, same MOA compounds, is probably the most relevant one. And the annotations are based on the literature and drug databases and so on, not based on the data. Okay, so some example results. So first of all, the similarity distributions change as a result of doing this embedding. This is an example cell line from L1000. You can see cosine of the known positive replicates is in gold. Cosine similarity of the background is in green, and the distributions are difficult to discriminate. With metric learning, the pairs of known similar signatures, the pairs of replicates, the distribution of similarity is in red, and the distribution of background similarities is in blue. And so you can kind of visually see, and we'll quantify, that PeML does a better job discriminating these two underlying distributions. And you see a similar trend in cell painting, that the distributions have fundamentally changed because of the embedding. Okay, so this is these next two slides kind of summarize the main results. So this is the first benchmark. We do cross-validation, so we train on 80% of the compounds and leave 20% of the compounds held out as test. We then evaluate on the test set the AUROC and the recall, the power, at a particular FTR. Each one of these rows represents a different cell line for L1000, and the cell painting data set is down here because it's all in one cell line. And so on the left, you can see the AUROC. Cosine has an average AUROC of around 0.72, whereas metric learning, it's closer to 0.85. So quite dramatic improvement, and in every context we look at. If you then look at the power to detect these signatures and discriminate them from the background, the recall for cosine is somewhere around 0.15, whereas for metric learning it's close to 0.4, 0.5. So again, quite dramatic improvement in the ability to identify these related signals. So replicates are kind of the space we're training on. If we look at the MOA, so now we're looking at pairs of compounds that belong to the same mechanism of action class. You can see that the AUROC with cosine is somewhere around 0.6, with metric learning is closer to 0.68. And the recall with cosine is somewhere between 0 and 0.1, whereas with metric learning it's closer to 0.2. And same MOA is, clearly it's a harder problem than replicate similarity. And there's sort of a lot of reasons why that might be the case. But the main bullet point is still that metric learning does a better job identifying these signals that we kind of expect to see. And finally, the mechanism of action, if you want to look at the signal to noise ratio, you can calculate that for metric learning, that's on the y-axis. Cosine is on the x-axis. Each one of these dots corresponds to one collection of compounds with the same MOA. So for example, this is compounds that are annotated to be estrogen receptor agonists measured in HA1E. And what you can see, like if you points above this y equals x line Metric learning does a better job discriminating them. So metric learning has improved performance, whereas below cosine is better. For the most part, most of these MOA classes are better discriminated with metric learning. There are a few that, for whatever reason, didn't embed very well. And you see the same thing with cell painting, that the metric learning signal to noise ratio is greater than cosine for essentially every compound mechanism of action class that we consider.
Can I ask a question, please? Can you please tell me exactly what is the prediction task for each individual compound? What are the negative samples?
Right. Yes. So for the signal to noise ratio, we compute the distribution of related pairs, of all related pairs, and we compute the similarity of everything else in the data set. And then compare the positive case are things that we expect to find, and the negative case is just everything else. And then you can ask, how well do you discriminate? Does that make sense?
Yes, I think so. But I don't know. I think wouldn't this, first of all, allow data leakage if you would apply your similarity metric on the same data set? So would it be wise to learn it on one data set and then apply it on a completely different one? But also, I think I have some other concerns, but I need to think about it a little bit.
Yeah, that's excuse me, that's a good question. Sorry. In the case of this result, we're doing cross-validation. So none of the examples that we're using were seen in training. In this case, compounds belonging to the same mechanism of action were never formally defined as positive pairs in the training process. Like the training process just uses pairs of replicates. I suppose it's possible that there could be some leakage or some sort of memorization, but metric learning doesn't know that two signatures belonging to the same MOA have any relationship at all.
Yeah, I was just confused because of the previous slide, you had two ground truths, and the bottom one said something like compounds of, yes, this one, compounds with the same identical mechanism of action.
Yes.
Yes, but doesn't it mean that they're different compounds, but just with the same mechanism of action? Then certainly there is some information exchange, no?
It's a question I'd like to think about. I mean, we in this task, we never compare pairs of signatures belonging to the same compounds. They're always different compounds. But yeah, I mean, certainly information leakage is a possibility when you're engineering a metric based on the data itself. But I'd like to think about that a little bit more. So please ask me afterwards.
Thank you.
Prudencio?
Yeah. So in the previous part of the talk, you were talking about the correlation between the number of dimensionality and the cosine, right? So I was just wondering if the comparison that you are doing here is on two vector size of the same size. The metric learning and the cosine are on the same vectors, same size of space, like.
Yes, we're the representation has the same number of dimensions as the original data. Obviously, you don't have to do that. Like if you were doing something with a VAE, you'd probably, employ some bottleneck and learn maybe, I don't know, a tenth of the latent features. But for this case, just because we're doing, essentially a rotation with skewness or with stretching along different axes, it's actually still on the same basis. It's just rotated somewhat. Does that make sense?
Yeah. Okay. Cool. Makes sense. Thank you.
But you're right. That probably wouldn't be fair. I mean, it'd be harder to sort of draw conclusions about that when the dimensionality is different. Okay, I'm running a bit over time, so I'll probably skip a few of these things. One thing I want to highlight is that it's sort of understood that the context of these experiments can be quite important because the biology of different cell contexts can be quite different based on the presence of mutations or tissue type and so on. So one question was, for the L1000 dataset where we have many different cell lines, is it better to learn a single metric for the entire dataset or to learn cell line specific metrics? So to do this, we trained a pan dataset metric, and then we trained a metric on each of about 20 different cell lines, and we're also comparing with cosine. And you can then evaluate this on the mechanism of action task. And what you see is that, first of all, both PeML similarity metrics show improved performance over cosine. So presumably we're learning something about the data. And also the cell-specific one, the context-specific metric, outperforms the pan dataset metric, which is probably not that surprising. Like, it's better if you get, like, you don't have as much training data if you're learning a context-specific model, but you're probably going to learn more relevant biology in your embedding. And you see the sort of same general trend with the recall task that the cell-specific models outperform pan dataset models, which in turn outperform cosine. I'm going to skip this part, but this is kind of asking how much data we need. Excuse me. We compared metric learning to cosine for all these results because it was kind of the natural null model. But I wanted to highlight that metric learning outperforms many of the other standard metrics like Pearson, Spearman, and a GSEA-based method, weighted CS, that, yeah, in essentially every cell line we looked at, metric learning has superior performance for mechanism of action recall. I'm going to skip the discussion of the eigenvalues. So just to summarize, I think this is a promising result. I definitely don't think that this is the final story in doing either weakly supervised learning or representation learning. But I think the takeaway, based on two different data modalities and two different data sets, is that biological analyses that involve, kind of unsupervised computing similarities among data points can be improved, potentially anyway, with metric learning. If you learn a representation, you can extract sort of more meaningful knowledge from it. And this has, of course, with perturbational analysis, there are some obvious things you might do, like try to identify mechanism of action, but there are lots of other things you might consider doing with data in this embedding, like clustering and classification and so on. I'd like to highlight that this is all possible because we have replicate experiments. Otherwise, like if you have a one-by-one data set where you measure each thing only once, you have no notion of whether two signatures are similar. So you really need data sets, and this is a good experimental design point, to always repeat experiments, because then you might be able to use that to learn some interesting embedding. Right, so I'm sorry, I'm running a little bit over time, but I want to mention a few future directions. I think the frontier for a lot of this representation learning is fully self-supervised learning. I want to highlight a paper, SUGPT, from the Bo Wang lab that trains a generative self-supervised learning model on single-cell RNA-seq data, a foundation model for sort of mining single-cell RNA, which looks quite interesting and quite promising. An open question in my mind is whether or not generative self-supervised approaches are more powerful than contrastive methods, especially for similarity metric learning. My intuition is that the contrastive architecture brings a lot in the sense that maybe you don't need as much data. I think a lot of the literature suggests that you can learn faster with contrastive approaches. And just the idea of doing repeated experiments seems advantageous to me compared to doing something like an attention mask that you have in these generative SSLs. But the holy grail in my mind for contrastive learning is identifying and finding these identity preserving transformations. If you could take an RNA-seq profile and apply a transformation that doesn't change the biological phenotype or the character of that, then you could do something like fully supervised contrastive learning. And I don't think something like adding Gaussian noise is going to be sufficiently interesting, because again, we live in some manifold, and so we want to find data points that are close on this manifold. There also are new perturbational data sets. The JUMP data set, or the JUMP consortium, which is spearheaded by Ann Carpenter's cell painting group at the Broad, recently released earlier this year a massive data set with over 130,000 unique perturbations. This is a really exciting resource for doing phenotypic screening. And also from the Carpenter lab, to your question earlier, Prudencio, Moshkov et al. Also uses weakly supervised learning specifically to do morphological representations and learn features from a convolutional neural network that better represent the underlying space, and they do some batch effect correction in their paper. So that's quite an interesting approach. Yeah. So to summarize, sorry, I ran quite a bit over where I expected, but sort of the thrust of my research is to learn representations on perturbational data that can help us discover more interesting things. We have a theoretical approach where we understand how cosine similarity depends on the data and how to find and identify interesting representations, the properties of interesting representations. And then perturbational metric learning is a very practical method for learning a new similarity function that, again, hopefully, and as far as the evidence I think shows, results in improved identification of biological relationships. And with that, thank you very much for your attention, and please let me know if you have any questions.
Perfect. Thank you so much, Jan, for the great talk. If you have any question, please just raise your hand and unmute yourself.
Yes, Dennis.
Yes. So this is again like a relatively basic question, but if you are learning your metric exclusively on the replicas as positive samples and everything else non-replicas as negative samples, how exactly do you regularize what happens to the negative samples? Because like you would think that it is possible that the metric will just learn only to basically collapse positive samples, and with the negative samples can happen anything anything possible. Of course, I can see that your metric is parameterized by a single matrix, right? So this does simplify things, but still.
It's a good question. Excuse me. I think it's helpful to consider the underlying theory from shoot, I seem to have lost my cursor. That one of the findings from the cosine distribution work is that there are limitations to how much you can kind of shatter your space. Like it's not. Let me back up and say that this in part motivated our choice of this loss function, this T loss, because it's actually not possible to have a cosine similarity distribution with negative mean. Like it won't go below 0. This is just the math. And so, it's also not possible with this approach to collapse all pairs of replicates to be the same point. Like if you have enough data points and your dimensionality is sufficiently low. I suppose if your dimensionality were sufficiently high, you probably could do that. But at least in practice, that hasn't proven to be an issue. But yeah, that is a bit of a diabolical case where, yeah, everything just collapses to 1 point. I'm actually in the self-supervised space. There is a whole set of generative approaches that don't do contrastive learning. They don't do positive versus negative, and somehow they avoid the collapsing case where everything collapses to a single point. And I don't completely understand that. But yeah, it is a realistic concern.
This was one of my concerns, right? So do you think that this works so well because the underlying data here is close to Gaussian? So essentially, you say that it is centered, but I have not recognized if it is necessarily normalized or not, but it is definitely not a count data, so.
Right, yeah. Yeah, I think Gaussian distributions are a reasonable approximation for a lot of perturbational data. I've done some other work that on L1000 specifically that highlights how deviations from normality actually can create a lot of problems, and the fact that the data isn't completely normally distributed makes it difficult. But yeah, with count data, yeah, any sort of discrete distribution, I don't really know how this would behave. So it probably helps a lot that the data is compact in a sense and lives in a nice little neighborhood.
Okay, thank you.
Paul?
One last question from Paul.
Yes, thank you so much. That was a very great talk, very clear. I have a question. So, of course, you're looking at perturbation data, but in other contexts, I'm interested in development or immunology or stuff like this. You could have dose response curve. And so is there a way you learn, a metric that will be common to something that is ramping up or something, like just more than perturbation, really like more like a continuous parameter that you will tune and learning something common to this to those perturbated system?
Yeah, that's a really interesting question. So my lab works in, see if I can find the relevant slide here. Yeah, my lab works in pharmacogenomics, where we measure viability and dose response curves and so on. And I mean, as long as your data lives in a vector space of some sort, it doesn't even necessarily right. It doesn't necessarily have to be a vector space, right? Because if you could replace these discrete features and the inner product with like an integration or something, KL divergence, I don't know, but some metric that compares continuous valued curves, and as long as you're still learning a similarity function, I mean, obviously you can discretize, right? If you have a drug dose response curve, you could sample doses and measure the viability and treat those as your features, and then do some sort of embedding. But yeah, I think continuous valued is certainly possible. I'd have to think about how to do it.
Okay, thank you.
Perfect. Thank you again, Jan, for the great talk, and thanks everyone for attending this one, and looking forward to see everyone next week. Bye.
Thank you very much.